Welcome to the BioPharma Business Intelligence Unit, where symbolic integrity meets strategic clarity. At BBIU, we go beyond the headlines to decode the structural forces behind global biopharma, AI, and economic realignment. If you value precision, verification, and symbolic depth, this channel is for you. Subscribe now to access our weekly briefings, dossiers, and intelligence reports, and consider joining our institutional tier to unlock early warning systems, custom metrics, and exclusive strategic insight. You're not just listening, you're entering the future of epistemic intelligence. www.biopharmabusinessintelligenceunit.com.
BBIU White Paper, From Enabler to Modulator, a Conceptual Framework for Enzyme-Controlled Subcutaneous Biologic Delivery. Abstract. Most current subcutaneous SC formulations using recombinant hyaluronidase, e.g., Halozyme's PH20 or Alteogen's ALP4, serve as transient permeability enhancers, enabling rapid delivery of biologics originally designed for intravenous IV administration. This article introduces a forward-looking concept, repurposing hyaluronidase not merely as a facilitator, but as a controlled-release modulator embedded within a biocompatible matrix. The goal, to regulate the spatial and temporal kinetics of antibody or bispecific delivery in the SC compartment without relying on slow-absorbing carriers or implantables. No experiments are claimed. This is a strategic design concept meant to trigger thought, caution, and opportunity. One background: what ALT-B4 and PH20 do today. Recombinant human hyaluronidase, rhPH20 or ALT-B4, temporarily breaks down hyaluronic acid in the extracellular matrix. Used to allow 10 milliliter subcutaneous injections for mAbs, ADCs, and fusion proteins. Enables faster systemic uptake, not prolonged release. Commercial success: Herceptin SC, HyQvia, Phesgo, and Turgase. But current systems are designed for convenience, not for kinetic control. 2. Conceptual shift: using hyaluronidase for time degradation. What if hyaluronidase is embedded in a matrix, such as cross-linked hyaluronic acid, polysaccharides, or synthetic gels? And instead of instant degradation, it acts progressively, controlled by enzyme concentration, pH, temperature, or co-released inhibitors. This would convert the subcutaneous space into a programmable biodepot, where release depends not only on diffusion, but on enzymatic erosion of the surrounding scaffold. 3. Application: antibodies, bispecifics, ADCs. Ideal for high-potency, low-volume payloads such as bispecifics or checkpoint inhibitors. Subcutaneous release curves could be tuned to match the desired pharmacokinetics. Fast ramp-up for cancer immunotherapy, flat steady state for endocrine treatments, pulsatile for neurology or rare diseases. This may reduce injection frequency or improve tissue tolerance. 4. Compatibility and feasibility. All components already exist: hyaluronic acid, hyaluronidase, and biologics. The novelty is in the integration and release control logic. The matrix can be temperature-sensitive, cross-linkable, or injectable in a two-phase format. Prior art likely exists from Halozyme, Genentech, or academic consortia, but most aim at permeability, not controlled erosion. 5. Strategic implication. This is not a patent proposal, but a positioning blueprint for biotech firms seeking subcutaneous options beyond volume increase. For investors, it suggests that next-generation subcutaneous formulations may be enzyme-driven, not just polymer-based. 6. Extended applications: small molecules, toxicity bands, and protective encapsulation. Most attention around subcutaneous delivery modulation has focused on biologics, antibodies, fusion proteins, ADCs. But the same principle could apply to small molecule drugs with narrow therapeutic indices. Use case: controlled digoxin release problem. Digoxin has a narrow therapeutic window. Rapid absorption or erratic release can push plasma levels into toxic ranges. Solution: embedding digoxin in a hyaluronic acid-based scaffold, shielded from enzymatic degradation, then co-administered with a precisely titrated dose of recombinant hyaluronidase, e.g. ALT-B4, could delay systemic entry, smoothen peak plasma concentration, extend half-life without repeated dosing. This approach could also apply to opioids, immunosuppressants, or chemotherapy agents, where stable release is more important than peak power. Protective layer function. The scaffold, e.g. cross-linked HA, provides a barrier against proteases, esterases, and nonspecific hydrolysis. This allows the core molecule, whether biologic or chemical, to stay intact until the matrix is gradually degraded by hyaluronidase. Conceptual update. Release is not only diffusion-dependent, but modulated by enzymatic erosion of the matrix, governed by hyaluronidase. SWOT analysis, enzyme-controlled SC delivery system. Strengths, versatile, applies to biologics and small molecules, uses well-known biocompatible components, enables kinetic control and molecular protection. Potential life cycle extension for mature drugs, compatible with current formulation technology. Weaknesses, not yet validated in vivo. Prior art may constrain patentability. Complex manufacturing and release tuning. Regulatory challenges for combo products. Opportunities. Reformulate narrow therapeutic index drugs like digoxin, chemo-opioids. Improve adherence via less frequent, safer dosing. Extend exclusivity via novel presentations. Fit for pediatrics, geriatrics, rural settings. Academic industry co-development potential. Threats. Existing patents from Halozyme, Genentech, and others. Competing delivery systems such as implants and microneedles. Risk of toxic release errors in high-risk drugs. Cost-effectiveness versus simpler SC formats. Adoption barriers including clinical inertia and regulation. Strategic note: this concept is shared as open knowledge for developers, clinicians, formulation scientists, and investors.